Hello and welcome everybody to my channel Omini Bioentrance, and today we will be discussing the Telangana SET question paper for the subject Life Science. This was conducted in the year 2024. So identify the sequence of events in the construction of a biosensor. Now biosensor is just like your glucose biosensor that you use to check for the sugar level in the patient or in a diabetic patient. That is just like a biosensor. So what are the sequence of events in the construction of the biosensor? So the first will be the entrapment of the enzyme. So you have to isolate the enzyme. Now for glucose biosensor, usually glucose oxidase is the enzyme that you isolate and you immobilize the enzyme in a particular matrix so that the enzyme will stay there intact. Then next is the cross linking. Cross linking, it will ensure the enzyme is functional and it is properly attached there, so that when you add the substrate for the enzyme, it will interact with the substrate and will give rise to a biochemical reaction that will be detected by the biosensor's detection mechanism. So that is with the case of enzyme reaction. So after cross-linking, after entrapment, you go for cross-linking, and then enzyme reaction. I told you add a specific substrate. So in the case of glucose biosensor, you add the blood of the diabetic patient. The glucose oxidase will act on the sugar present in the blood of the diabetic patient, and it will give a biochemical reaction that will be detected by the biosensors detector. Then will be the response, that is it will tell us regarding the concentration of the analyte in your sample. Okay, so that is the steps involved in the construction of the biosensor. So first it is entrapment, then cross-linking, then enzyme reaction, and then response. Then next one, given below are some terms and concepts related to phytoremediation in column A and B. Now you have to match these. So first is what is phytoremediation? It is a method of cleaning up your environment of heavy metals or other pesticide or insecticide by using plants. So these plants, they will either remove or degrade or stabilize the pollutant in the soil, water or air. So what are excluders? Excluders means they can resist, they can restrict the heavy metal in their roots, and thereby they will prevent the metal from being translocated to other parts of the plant, and they will detoxify them by restricting them in their roots. So A is 4, can restrict heavy metal ions to the roots and detoxify them. Then heavy metal transporter protein, it is with regard to transgenesis. So heavy metal transporters are plants involved in the uptake, translocation, then storage of heavy metal from the soil. And in transgenics, if you transform a plant with the gene encoding for these transporters, then the plant will develop the heavy metal accumulation capacity and will become more efficient in phytoremediation. So B will be 3. Then hyperaccumulators means they are plants that can absorb, translocate the heavy metal to different parts of the plant, and they will not show any toxic symptoms of the heavy metal. They are called as hyperaccumulators, and they are also involved in the process of phytochemistry. remediation. So can absorb and transfer heavy metal to aerial parts without phytotoxicity. So C will be 1. Then high biomass non-accumulators. High biomass means they have higher growth rate. So high biomass, but they will not accumulate any pollutants in their tissues. So they can also be used in phytoremediation. So here they have given 2 examples of high biomass non-accumulators. They are the Salix and the Populus species. So that is D2. So the answer is option 2. Then next one, identify the incorrect statement. 2,4-D or 2,4-dichlorophenoxyacetic acid is the most commonly used chemical analog of indole 3-acetic acid or IAA. Now 2,4-D, it is a synthetic auxin, and IAA is the naturally occurring one. So 2,4-D is a chemical analog of IAA. That is correct statement, but that is not our answer. Then in somatic embryogenesis, embryo initiation needs high concentration of 2,4-D. Yes, to initiate embryo from somatic cells, usually you add high concentration of 2,4-D. Then crown gall disease depends on the presence of TI plasmid in Agrobacterium tumefaciens. That statement also is correct. Agrobacterium tumefaciens have got the TI plasmid or the tumor inducing plasmid, and this plasmid has got a region called as the tDNA, which is introduced into the plant cell when the plant cell gets infected with the Agrobacterium tumefaciens. This will tDNA, it will go and integrate into the plant genome, leading to uncontrolled growth, which is called as GALS, G-A-L-L. Hence this statement is correct. Then Agrobacterium tumefaciens is responsible for hairy root formation in plants. That is not correct, because it is Agrobacterium rhizogenes which is responsible for hairy root formation in plants. And Agrobacterium rhizogenes, it is having the RI plasmid which is causing this hairy root formation in plants. So 4 is the incorrect statement, and that is your answer. Then match the group 1 with group 2. So insect resistant cotton, how you developed? It was by using the cry gene present in the Bacillus thuringiensis or the BT gene. Okay, BT gene of Bacillus thuringiensis. It was introduced into the cotton plant. This BT gene, it produced a protein which was harmful for the insect which attacked cotton. And by introducing this BT gene, you got a cotton which was resistant to this insect. That is the cotton ball worm. That is the insect. So A1. Then golden rice, it is for it was developed or engineered to produce beta carotene to face the vitamin A deficiency, because beta carotene is a precursor of vitamin A. So B3. Then flower saver tomato, it was developed for extending the shelf life of tomato. So what they did was they silenced the polygalacturonase gene, and this polygalacturonase gene, it was responsible for fruit softening, which happens during ripening of the tomato. So when this particular gene was silenced, the tomatoes had extended shelf life and they remained more firmer. Then herbicide tolerance, glyphosate. So glyphosate, it is a herbicide. It was widely used, but on application we found that the scientist found that glyphosate will also harm the crop plant, not only the weeds. So what they did, they genetically modified the crop plant to become resistant to glyphosate, and they allowed this resistant transgenic plant to grow in the field. Now the farmers can happily use the herbicide, that is glyphosate, to control the weeds, because now the weeds are not resistant to glyphosate, so they will die off, and the plant which is herbicide which is tolerant to glyphosate will survive upon spraying with glyphosate. So D is 4. Then next one, as cancer progresses, several genome rearrangements including translocation, deletion, duplication, etc. occur. If these rearrangements are to be identified, which of the following technique would be most suitable? It is the multicolor fish or multicolor fluorescence in situ hybridization technique. So here this technique, it uses the fluorescent labeled probes that target specific DNA sequences on the chromosomes, and they will, as a result, you can visualize these chromosomal aberration like deletion, duplication, translocation under the microscope, and it will show these changes under very high resolution. So 3 is the answer. And flow cytometry, it is mainly used for analyzing the cell surface markers. That is, in case of your immunology, you have got the CD4 plus CD8 plus markers. All these can be analyzed by flow cytometry. Then RAPD, it is for detecting genetic variation between two individuals, not for translocation, duplication, deletion or chromosomal arrangements. Then microarray, yes, it can detect duplication and deletion, because it will result in change in the copy number, but they are not efficient for detecting translocations. Okay, so microarray is not the answer. So answer is multicolor fish. Then next one, resolving power of a microscope is a function of. So resolving power, it is given by the formula 0.61 lambda, which is wavelength, and Na, that is numerical aperture. So wavelength of light used and numerical aperture of the lens system. So that is the resolving power of microscope. Then which among is the cell line of human embryogenic lungs? It is MRC5. Answer is option 2. Then next one, page is used to separate protein fragments based on size. Yes. Now how does page separate these protein fragments based on size? Because the gel matrix of the page, it is polyacrylamide gel. So this gel matrix, it will act as a sieve. So the proteins with the smaller size, they will migrate faster in the gel, whereas the larger proteins, their movement is slower. So based on the difference in the size, you will get different bands in a page gel. So what is the reason they have given? When current is applied, ions separate out and migrate towards the attractive electrode. That statement is true, but it doesn't explain the reason why the page is separating protein based on size. It is a general statement for any electrophoresis. When current is applied, ions will separate, migrate to the opposite electrodes. That is correct for electrophoresis technique, but not why the page separate protein fragments based on size. So here both assertion and reason are true, but reason is not the correct explanation of assertion. Then next one, here are the biostatistic terms, and you have to, or the test types and the concept why they are used. So Crucial-Wallis test, it is a non-parametric test to compare means of more than two population groups. So A is 2. Then Z test, that is for testing the difference between means of two sample groups. So B4. ANOVA, again a parametric test to compare means of more than two population groups, and chi-square, it is for goodness of fit. So it is a non-parametric test to test the goodness of fit. All these terms and why these tests are used, you have to know. And next one, amplified fragment length polymorphism. So what are the steps involved in an AFLP? So first you extract the DNA, then you digest the DNA with the suitable restriction enzyme, then you ligate adapters to the restriction fragments. Adapters means they are short double-stranded oligonucleotides. So these are ligated to the ends of the restriction fragment. Then you selectively amplify some of these fragments with PCR. So when you are conducting PCR, you will design primers for the adapters. Okay. And you do the amplification by PCR. Then you separate them with the electrophoresis and visualize the markers and photograph the gel, etc., etc. So here is a PCR based tool. Yes, you are using PCR there. So P is correct. Uses restriction enzyme to cut genomic DNA. Yes. Involve ligation of adapters to the sticky ends of the restriction fragments. Yes. Involves amplification using primers complementary to the adapter. That is also correct. So P, Q, R, S all are correct. Then next one, freshly cut prepared culture, freshly prepared from isolated tissue is known as primary culture. So primary culture means suppose you want to have the primary culture of the liver cells of a rat. So you take the rat, then dissect it, isolate the liver. From the liver you isolate the liver cells and you grow them in a culture medium. So that is primary culture. It is freshly prepared. So directly isolated from the tissue or the organ and grown in artificial medium. So that is primary culture. Then organ culture means it is a laboratory technique for growth, maintenance of whole organs or organ primordia or small fragments of the organ. So that is not a culture of freshly prepared isolated tissue. All that it is not correct. Then cell line means they are derived from the primary cultures and they are adapted to grow in the culture medium or in vitro conditions. That is cell line. Then histotypic culture means growing a single defined cell type. A single defined cell type is grown at high density on a three-dimensional matrix. So this three-dimensional matrix can be a sponge or a gel, and this histotypic culture, it will result in the formation of tissue like structures. So here answer is option 2, primary culture. Then next one, the last question, that is X-ray crystallography. It is based on elastic scattering of X-rays. Yes. Non-destructive analytical technique. Yes. The sample is not destructed after analysis using the X-ray. Okay, the sample is not destructive, but if you use techniques like what is that? Oh, I'm not. Mass spectrometry. There is chances that you ionize the sample in mass spectrometry, so it will cause a destruction to the sample. But here that is not happening in case of X-ray crystallography. Okay, that is what it is meant by a non-destructive analytical technique. Then used in determining the arrangement of atoms within a crystal. Yes. For three-dimensional arrangement of atoms in the crystal can be determined using X-ray crystallography, and it is based on the Bragg's law. So P, Q, R, S all statements are correct. So with this I have finished the whole question paper. Thank you for watching my channel. Please do like, share and subscribe. If you have any comments, you can give it in the comment section. Thank you.